Hello, welcome to Module 5, Chapter 9. This is muscles and muscle tissue. And this video, we're going to talk about the sliding filament theory of muscle contraction. So in this cartoon from Shutterstock is a sarcomere. We've talked about that in a previous video. But a sarcomere goes from, it shows you here, goes from Z line to Z line. And the red filaments in this cartoon are actin, and they're the thin filaments, and then the blue filaments are the thick filaments. So the thin filament is made of the protein actin, the thick filament is made of the protein myosin. And this shows that when the sarcomere shortens, so you can see the top cartoon, the top part of the cartoon, these myosin heads are here. And then remember, there's myosin head binding sites on actin. So when the muscle is stimulated, so that means that there's an action potential sent from a motor neuron to the neuromuscular junction, and then you get the biochemistry across the neuromuscular junction. You send an action potential down the muscle cell plasma membrane or sarcolemma. That action potential dives into the T-tubules, and then more biochemistry happens, and the sarcomeres get shorter. If you have thousands of thousands of sarcomeres getting shorter in the length of a muscle, in your whole muscle, then the whole muscle is going to get shorter. So these sarcomeres, upon getting a signal to get short, do so. So the top part of the cartoon shows a sarcomere at rest, and then the bottom part of the cartoon shows a sarcomere in a contracted muscle. And you can see the two Z lines have moved closer to each other, and what happened doesn't show it in the cartoon because it's not a video cartoon, but the myosin heads have basically walked along the myosin head binding sites on actin, thereby pulling the two Z lines closer together. So in this cartoon, we've talked about this before. This is a graph of an action potential. So here's the voltage in millivolts. Here's the time in milliseconds. And it starts off with a resting membrane potential of about 70 millivolts. So resting membrane potential, and then you get a stimulus, so maybe a graded potential with some sodium coming in via ligand-gated sodium channels. And if enough sodium gets in to reach threshold potential at -55, then the voltage-gated sodium channels open. You get depolarization of the plasma membrane and repolarization, and back to the resting state. That's an action potential. So the action potential travels from the neuromuscular junction. So this is a cartoon of the neuromuscular junction. We did a video on that. So this is an axon terminal in yellow. Here's the folds of the phospholipid bilayer of the muscle cell. And you go from the axon terminal, if you remember, you release acetylcholine. It opens the ligand-gated sodium channels. Sodium rushes in, the inside of the cell becomes more positive, and then that will open the voltage-gated sodium channels here just outside the neuromuscular junction, and then you can fire an action potential along the whole length of the phospholipid bilayer of the muscle cell. And then it'll dive into, I have a cartoon hopefully, that action potential will dive into the T-tubules of the muscle cell, and then biochemistry will happen. Calcium will be released into the muscle cell, more biochemistry, and you get contraction. So in the T-tubules of the muscle cell, there are voltage-gated calcium channels, and there's also ligand-gated calcium channels on the SR, but through magical biochemistry and the action potential diving into the T-tubules, the voltage-gated calcium channels will open, and then the ligand-gated calcium channels will also open in the sarcoplasmic reticulum, and calcium will flood into the intracellular fluid. Oh, there's my cartoon. Thank you. Should have put that up sooner. But so here's these white things. These are the T tubules. And here's the sarcolemma, aka the phospholipid bilayer. So as the action potential travels along the phospholipid bilayer, muscle physiology, the sarcolemma, that action potential will dive down into these T tubules, and it will change the membrane potential. So voltage-gated calcium channels in the T tubules will sense that change in voltage that the action potential brought to the T tubules, and that will open voltage-gated calcium channels. Calcium will rush into the muscle cell from the outside of the cell to the inside of the cell, and then there'll also be that calcium will also activate calcium-gated or ligand-gated calcium channels that will release calcium from the SR. So you've got calcium coming in from the extracellular fluid, you've got calcium coming in from the SR. So the whole inside of this muscle cell or fiber is going to be flooded with calcium. So calcium's going to do something important, and we'll see what that is with the sliding filament theory. So once inside the muscle cell, the calcium will bind to these regulatory proteins on thin filaments. So here's our cartoon. So this orange thing, that's a thick filament or made of the protein myosin, and here's a myosin head. And then these purple spheres are actin, G-actin for globular actin. And then you have these structures. So it looks like a kind of a orange-yellow rope, and then you have these little circles, these little yellow circles. So these structures, the yellow circles and the kind of the ropey-looking yellow thing, that's the troponin tropomyosin complex. And when the muscle cell is at rest, these myosin binding sites on actin are covered by this tropomyosin troponin complex. But when calcium is present, calcium can bind to this regular protein, troponin, and it will move the troponin tropomyosin complex out of the way. Now myosin head binding sites on actin are exposed, and myosin heads can now bind to the myosin head binding sites on actin. And then through a series of biochemical events, these myosin heads can bind and they can detach, recock their heads, and move the sarcomere, the two Z lines of the sarcomere, together. And it says here, so the attached, once the myosin heads are attached, they pivot, and they're going to slide the thin filament toward the center of the sarcomere. And then that's how you get the sarcomere shortened and you get contraction. So this part, it says number 2, is the working stroke of the myosin head. So the myosin head will pivot and bend. So you can see how it's different. So look at the myosin head here and look at the myosin head here. So at this stage, the myosin head has pivoted and bent, and it pulls the actin filament toward the M line. So I don't know if you remember when we talked about sarcomeres in a previous video, but there's a H zone, and in the middle of the H zone there's an M line, and the actin filaments slide toward the M line, making the sarcomere shorter. That's why they call it the sliding filament. And then you have to have ATP present in the muscle. As long as ATP is there and calcium is present, so that if calcium is present, then the troponin tropomyosin complex is not covering the myosin head binding sites on actin. And if you have ATP, ATP actually allows the myosin head to detach. So if ATP is not present, the myosin head would stay stuck to the myosin binding site on actin. So here's the whole thing together. So it starts at this point. Number 1 says the myosin cross bridge attaches to the actin myofilament, and you can see I've got calcium here. I don't know if you can see that, it's pretty small, but you've got calcium here. So calcium has removed the troponin tropomyosin complex off the myosin head binding sites on actin, And then number 2, it says this is the working stroke. So the myosin head pivots and bends, and it pulls the actin toward the midline. So I've used this the Shutterstock cartoon up here. So the actin, as the myosin heads on myosin bind to the myosin binding sites on actin and move, and that the myosin heads pivot and move, that's going to bring these actin filaments closer to the M line. So here's the H zone and the M line. And the same thing as the actin filaments on this side. So as these myosin heads are forming attachments and detaching, they're doing this kind of working stroke thing that pulls the actin toward the midline or the M line of the sarcomere. And in order for the myosin head to actually detach from the myosin head binding site on actin, it needs ATP. Okay. To end the contraction, there's the action potential has stopped to the muscle, and then calcium, using calcium pumps, calcium gets pumped back into the SR, and can you guess what happens when there's no more calcium inside the cell? If there's no calcium, take a guess. If you said that when there is no calcium inside the cell, then the troponin tropomyosin complex moves back over the myosin head binding sites on actin, you'd be right. So this just gives a little more details on what myosin looks like up close. This is a model of myosin. So it looks like 2 golf clubs intertwined. So here's the myosin head. Okay, and then this is just another cartoon of the myosin protein. Here's the myosin heads, here's the M line, and then this guy. And the center here without any myosin head, that's the H zone. This is a nice cartoon showing the thin filament made of the protein actin. So here's actin. It's actually G-actin because it's in these little globule forms. Here's tropomyosin. Here's troponin in yellow. And remember, when calcium is present inside the cell, it's going to bind to this troponin complex. And then the troponin tropomyosin complex can move out of the way of the myosin head binding sites on actin. And you can see that in this cartoon. So this is a cartoon that kind of shows the whole story, starting with the neuromuscular junction. So here's the motor neuron. Here's the axon terminal of the motor neuron. Remember, the action potential comes down the axon of the motor neuron, and it opens these voltage-gated calcium channels, and then calcium floods in, a whole bunch of biochemistry occurs, and then these acetylcholine-containing vesicles in the next step, the acetylcholine-containing vesicles will release acetylcholine into the synaptic cleft, and then the arrow comes over here, and then, so this is the sarcolemma of the muscle, so the phospholipid bilayer of the muscle. So acetylcholine binds to these ligand-gated sodium channels, and they're actually sodium and potassium channels. So you could also just call this a ligand-gated ion channel, and with the ligand being acetylcholine. Sodium rushes in, so it makes the inside of the muscle cell more positive. And in this cartoon, in this book, they call this, because it's muscle physiology, they call it an end plate potential, but it's also called a local potential. Okay, then step 5 comes over here. When enough sodium has come in via the ligand-gated sodium channels, enough positive charges have built up inside the cell, then these voltage-gated sodium and potassium channels will open, and now you can start an action potential because there's voltage-gated sodium and potassium channels all the way down the phospholipid bilayer. And then, so the action potentials in yellow here, the action potentials are propagated, or they go down into the T tubules, and then there are voltage-gated calcium channels in the T tubules, and then there's calcium-gated calcium channels in the SR here. but they both will release calcium into the intracellular fluid of the muscle. And then you come over here to number 8, calcium binds troponin, and then the troponin-tropomyosin complex will, upon receiving the signal from calcium, will move out of the way of the myosin head binding sites on actin. And then the myosin heads, once the myosin head binding sites on actin are exposed, then myosin heads can bind to the myosin head binding sites. They can move, so you can see in this cartoon they're moving. The myosin heads move, and what that movement does is it slides actin past the myosin filament. So actin moves toward the M line of the sarcomere, and the whole sarcomere gets shortened. And this just shows all the different parts of that myosin head binding. So there's formation of the myosin actin cross bridge here when they're linked, and then the power stroke happens when the head pivots, and then when ATP binds, the head can release and go find a new attachment site on actin. And then step 14, cessation of nervous stimulation. So that means there's no more action potential. That means there's no more acetylcholine in the synaptic cleft. Then acetylcholine esterase is an enzyme, can break down acetylcholine that's there, and then they'll put parts of that acetylcholine back into the axon terminal. And then calcium also gets pumped back out of the cell, and calcium also gets pumped back into the SR. When there's no more calcium present, then troponin-tropomyosin complex moves over the myosin head binding sites on actin, and myosin heads can't find a binding site anymore. And so the muscle contraction stops. And this is just another big picture that helps. This is from Marieb. You can read over this. It talks about what I just talked about. Oh, but you know, I remembered why I like this cartoon, because in the top here, you can see the T-tubule. And it says it shows a calcium release channel on the SR. So there's voltage gated calcium channels in the T-tubule, and there's calcium gated channels on the SR. Anyway, that's a big picture.